Okay, so this is um so yeah, I just did present at ARVO. So this is a very like a separate project from that we have been working on, and it's been data we collected a while ago, and we're just working on following it up and taking two different approaches to follow it up. So as I said, I'm very interested to get some feedback um, on just different aspects of it. And the main focus of it, as you'll see, is how the RPE are responding to serine and glycine. So for those of you who don't know me, my name is Mari Gantner, and I am a scientist at the Lowy Medical Research Institute. And the Lowy Medical Research Institute is a private funded institute, and our main goal is to study and understand the disease macular telangiectasia. So this is a retinal specific disease that has a moderate prevalence, would be considered maybe semi-rare, 1 to 10,000, and the age of onset is in the 40s and 50s, and it's specifically the macula that is most affected, and that leads to progressive loss of central vision in the patients with MacTel. So from post-mortem histology, we've gained some insight as to what is actually happening. And in the MacTel region, or the macular zone, what is observed first is this loss of Mueller cells, and then that appears to be followed by rods and then later cones. And that is all very tightly contained to this MacTel zone. Interestingly enough, what was observed in the 4 different postmortem eyes that have been examined so far is that there does seem to be a pan-retinal RPE defect. And what is observed across the retina, extending outside of the macula, is a buildup of debris between the RPE and the photoreceptors. And also, I'm not showing an image here, but there also appears to be lack of phagosomes inside of the MacTel patient RPE cells. So what's somewhat nice about this pan-retinal defect is this is something we think we might be able to model in a mouse which don't have macula. So we're not expecting necessarily to see this distinct macular defect that the patients get, but maybe we can model some of these more pan-retinal defects. So MacTel has a genetic inheritance, or a complex genetic inheritance. So it is not a monogenic disease. In over the 4,000 patients that have been seen so far and sequenced, about only 4,000 of them, or 4% of them, present to be driven by rare coding variants in particular genes. And the vast majority of the patients then seem to be driven by either multi-factors or non-coding and less rare genetic drivers. Despite that complexity, though, we still got a lot of information from the genetics, because a lot of the hits, both with the rare coding variants and then also the loci that are found to be associated with MacTel risk, lie near or in genes that are associated with serine and glycine metabolism. And this is just some of the key ones that have been seen so far. So it's how serine and glycine are sourced and also how they're disposed. And what's interesting, following up with the genetics or validating this idea that serine and glycine might be central to this disease, is when the serum from MacTel patients is looked at from global metabolomics perspective and compared to controls. There are a lot of metabolites that are significantly different between MacTel's controls at a systemic level, which is fascinating because it's a disease that we think just impacts, or historically was thought to just impact the macula, but it has this clear metabolic systemic signature. And 2 of the most significantly affected metabolites are serine and glycine, and these are both reduced in the serum of MacTel patients. So they have a diverse genetic signature that points to serine and glycine, and then this common reduction of serine and glycine systemically. So I'm not going to really go down the pathways of serine and glycine, but I did want to point out that serine and glycine do much more than just build amino acids or just build proteins. These 2 amino acids are tightly connected to a lot of different metabolic pathways. as I'll talk about in a little bit, they're connected to glucose metabolism, and then they themselves are the building blocks to a lot of different molecules, and also the conversion between them is what fuels the folate cycle and feeds into the methionine cycle. So their levels being low could have a lot of implications. To model the low serine and glycine that we're seeing in our patients, we decided to take an approach, instead of a genetic approach, to take a diet approach and make a custom diet that lacked serine and glycine and feed this to mice. And in that way, we could systemically reduce the serine and glycine, which we think reflects this broad genetic drivers having a common metabolic signature. And so when we do this, we put C57 black 6 mice either on a control diet or a serine glycine free diet. Within a couple days, we see that the metabolites are already affected, and this is in the plasma and the retinal tissue. We see serine reduced, so this is showing actually a circadian rhythm for the serine. It's reduced in the blood of the mice. Glycine is even further reduced, and if we catch them in a fed state, the serine and glycine levels are also down in the retina in the back of the eye. And the lipid changes that we associate with this are also seen within 1 week. Interestingly, when we've put these mice on this diet and then watched them over time, looking to see if there's any consequence to the eye, it takes almost a full year before we start to see a retinal defect. And the first thing that presented was an ERG defect. And what we saw was a photopic B wave reduction, so suggesting that the cone pathways are impaired. As the mice get older, we see even more aspects of the ERGs disrupted, but this is the first and the most consistently reduced phenotype we see in the mice. So what
I'm sorry,
we're
Mari.
now, yeah.
When in patients, have people determined whether like some of the early defects that might happen with one of these mutations in like their ERG?
So there have been, so according to the clinicians, the ERG is not a common problem that's seen with the MacTel patients. In more careful studies recently, where they're doing this dark adapted ERGs, they're actually seeing that there is a defect, and it seems to be more rod than cone, that extends outside of the macular region. That's worse towards the macula, but still is there outside of it. So we do think that there could be some ERG defect that is underappreciated by the way it's being measured currently. And so they're following up on that. That answers your question. Okay. So given that it took a while for the phenotype, but the metabolites seemed to change so quickly, we were interested in kind of looking at what's happening in between that, and can we get some insight into the cell functions that are being changed. So we decided to do an RNA-seq of the mice after we see the metabolites altered, but well before we see the phenotype, to see if we could see the early things that might be leading and driving the phenotype. So we started with bulk RNA-seq, and we took mice fed the control diet or the serine glycine free diet, and we separated out the neural retina from the what we call the back of the eye, or the RPE choroid, and then we ran the bulk RNA-seq on those separately. And so from what you can see in this MDS plot, which is similar to a PCA, which is looking how the samples relate to each other, when we look at the neural retina specifically on the serine glycine free diet versus the control diet, there's little distinction between what the diet does to the transcriptional profile in those tissues. However, when we look at the RPE choroid, there's a huge distinction between the RPE choroid of the diet animals and the serine glycine free fed, or the control diet and the serine glycine fed diet. And it's almost like the same distance as the RPE from the retina. You see the RPE is changed by this diet. So this was very dramatic. And it's also reflected in the number of genes that are significantly impacted. So the differentially expressed genes in the retina, we saw in the total retina, we saw only about 20 genes that were differentially expressed, where in the RPE choroid tissue, there were nearly 1,300 genes that were differentially expressed. So a huge proportion of the RPE choroid's transcriptome was altered by this several month feeding of the serine glycine free diet. So simultaneously, or a little bit later than the bulk RNA-seq, we also were doing some single nuclei RNA-seq, and we did this in the diet animals as well. But what we did was we combined the retinal tissue and the RPE choroid into one sample. It was quite expensive to run these, so we only did N of 2, and we decided to combine tissue. And to try to figure out which cell type is responsible for that bulk signal, we looked at our single nuclei data. And what we found, let me actually see if I can get my cursor working, was that when we combined the neural retina in the back of the eye and ran the single nuclei RNA-seq, there were 4 cell types that appeared that were clearly from the back of the eye and not the neural retina. So we got RPE stromal cells, endothelial cells, and melanocytes. And looking at just those cell types that were derived from the back of the eye, we found that within those cell types, the RPE were really the only cell type that had a significant change in the number of genes expressed. So based on this, we really think that the signal in the bulk is coming from the RPE cells as opposed to the other cell types in the back of the eye. So our single nuclei, we only had N of 2, and we couldn't really, it wasn't a very deep sequencing, and we didn't have the power to do much statistics. So we actually returned to the bulk RNA where we had that huge data set. And to start to understand what's in that data set, we did pathway enrichment analysis. So we just looked at across all of the genes, what directionalities were they and what pathways were hit. And so what we found when we did this was, I think we had over 100 pathways that are altered, but the top 30 clustered into kind of some distinct groups. Extracellular environmental sensing is what we called this one group, central carbon and lipid metabolism, and then cell signaling, mostly related to neuronal signaling, was the third group. But when we looked closely in this top group, what caught our attention was phagosome and lysosome. And that's especially with this idea that we think we're looking at changes in the RPE. And as most people in this group appreciate, one of the main functions of the RPE is to chew up and recycle the spent outer segments of the photoreceptors. So we were curious in following this up to see if there was a potential defect that the low serine and glycine were causing to this function. And especially given what had been observed in the MacTel patients, that there's this buildup of debris between the photoreceptors and the RPE, and then also a loss of phagosomes visible within the RPE themselves of the MacTel patients. We thought this was worth exploring. So we we used an in vivo assay where we took the eye cup from the mice, made a flat mount with the RPE facing up, and then stained that for ZO1 to get our borders and rhodopsin. And this is an assay that we adapted from Claudio Punzo's lab, was one of the times I had seen this presented. And so we used, he helped guide us on this. It's the phagocytosis of the outer segments is a circadian regulated process. And if you look at light onset, that's when you're going to see the largest number of puncti stained, rhodopsin positive puncti stained on the RPE. And then as the day goes on, that number will decrease probably, or the idea is because they're being degraded, being taken up and degraded as the day goes on. So we did this in our control and our serine glycine free fed animals. And we looked at 11 a.m. We looked at a couple different time points, but I'm showing 11 a.m. here because this is like supposed to be like within that kinetic window where you can catch a lot of differences. And what we saw was that there was a very clear increase in the number of rhodopsin puncta in our serine glycine fed animals, suggesting that maybe the outer segments were not getting taken in and degraded as they were in the controls. We also looked earlier, closer to our light onset, and saw a trend, but not the same significance as when we waited later in the day. So this was kind of giving us our first hint that there was something going on, and we're still following up on this. And Jason Miller, I talked to him a while ago, and he had told me about an assay where to look at the degradation of rhodopsin in photoreceptors will give you an idea of how the phagocytosis is progressing. So our flat mounts is kind of showing us that maybe they're not getting in well, but we don't know, is that because they're not chewing them up well? So we took the back of the eye tissue from the mice, either control or serine glycine free fed, and we ran a Western for that, looking at the degradation pattern of rhodopsin. And so what we did notice strikingly, so when we dissected these, we did one control, one serine glycine, one control, one serine glycine, and alternated, because in that 11 a.m. window, things are changing quite rapidly. And it was really the timing of the dissections, even though we were going super fast, like 3 minutes per eye. I think the biggest change we can appreciate is that during that time we were dissecting, we were seeing loss of or degradation of rhodopsin. But there wasn't a striking difference. Given that variability of the time, we couldn't appreciate a striking difference between the serine and glycine and controls. So while we don't feel confident saying that there's no problem with degradation, at least in our hands, it doesn't appear to be a striking loss of the ability of the RPE to degrade the rhodopsin that is coming in.
Mari, so
Yeah.
this is Jason.
Yeah.
So when we're looking at these are all, just to clarify, these are all about the same time point, so they should sort of be complete replicates of each other, but the dissection goes from left to right in terms of you started with that animal and the right one is the last animal on the
Yeah, so I
control.
started with this control animal, then I did this minus SG
Yeah.
animal,
Okay.
or I might have started even SG, then control. I alternated one of each, and the left to right is the order in which I did them.
But they're all theoretically the same time point, like you said, except that it's whatever the time it took to do the dissections.
Exactly.
Got it. Okay. Thanks.
Exactly. Yeah. I think I did them all within like 30 minutes. So it was shocking that there was this big of a difference. I know you've done this in cells. Have you done this in vitro or in vivo?
Animals are too hard.
Okay.
I'm just joking.
Because I'm wondering if
I'm
this
just
is a
joking.
common.
I don't know if someone's actually been as careful as you in keeping track of the temporal order of it. I think that's super interesting. I'd be curious.
Yeah, we were pretty bummed because it just made it really hard to, like, if we have a subtle, unless it's a huge defect, I don't think we're going to be able to detect it. And for our flat mounts, we ran into the same problem with these, that, if it took 30 minutes, the one that you collected at 11 versus the one you collected 11:30 showed a big difference. So we started just doing 2 animals a day, always at 11, and being done within like 8 minutes, like really trying to stick very tightly to that time point. So yeah, but yeah, we think if it was something, it might be more striking than this. There's potentially like something more holding on, but it seems too variable to really call it. So it's kind of an unknown. So we decided mice are hard, so maybe we would try some cells. So we worked with the Lonza human fetal RPE, and we differentiate these for over 2 weeks in low FBS media and get nice differentiation. And then we designed a custom media working with a different company that is like the Lonza media, but we were able to design it so that it lacked serine and glycine, and also glucose, and other things that we would want to use for tracers. So after the cells were well differentiated, then we put them for varying amounts of days or weeks into a serine glycine free media. And to look at this phagocytic function, we used porcine photoreceptor outer segments that we had isolated and then labeled, and added these to the cells for 5 hours. And one of our assays was then to trypsinize the cells, get them into a single cell suspension, and look at flow cytometry to look at the uptake of these labeled outer or outer segments. We also did some just rinse the media off, rinse the unbound outer segments off, and did some staining right there in the dish. And I want to get onto the metabolism part of this project. So I'm not showing the flow cytometry data, but what we did see was that there was a subtle but very reliable decrease in the number of POS that were internalized in the cells. But what we think we got the most insight from was this cell staining. And I'm regretting we don't have the ZO1 in here, but this is a picture of the RPE cells taken from above. And we actually just stained for rhodopsins. These were unlabeled outer segments that we had put on for 5 hours and then rinsed off a lot with PBS to try to get everything that's not bound rinsed off. So you can see this is control media. This is a serine glycine free media for 2 weeks. And then this was control media, but an inhibitor added for 5 hours. So for the time that the outer segments were added. And Dinosaur is an inhibitor of the dynamin GTPase. So it blocks the final pinching off of the endosome. So what we found was that, kind of similar to what we had seen in the flow cytometry, is that there were fewer internalized POS, or these outer segments, on the serine glycine free cells. And because in this case, the Dinosaur actually showed a lot more, we think it's not an issue with the actual engulfment of the outer segments and bringing it in. We think it's above that step. So it's the binding and recognition and the actual ability to hold the outer segments down that would be impaired in our serine glycine free media. So I'm trying to think of what, yeah. So kind of, so in the, to summarize, like in the in vivo, we saw this buildup of outer segments on top of the RPE. And then in a situation where we had them in vitro and we were able to really wash, they seemed to wash away. So we think that there is maybe a binding issue as a consequence of the serine and glycine. So to summarize this part,
Mari, I was just,
yeah,
this is Jason again. Just, I think a complementary experiment for that is to do the same phagocytosis analysis, but when you fix, don't do any detergents. So then you'll really know if it's truly cell surface or if it's just internalized, which can help I mean, then you can know whether it's truly a binding or if it's an early initiation.
yeah, that's a great idea. And we've actually, Caleb, who's on this talk right now, he's one who's put a lot of work into this, and those flat mounts. He's tried that within the in vivo flat mounts in the RPE, where he did one rhodopsin antibody before permeabilization, permeabilize, and then do a different rhodopsin antibody, and hoping that we could see and distinguish, you know, these ones sitting on top versus the ones that are inside. And that didn't give us consistent results in vivo, but we haven't tried. It's a good point. The in vitro, these cells are so much easier to work with in a sense. We should try a similar assay in here, and that would really help. That's a great idea. Distinguish.
I mean, just to clarify, you know, it's not a perfect, because PFA itself can permeabilize. So you know, so it's not like it's, I mean, it theoretically sounds great, but you'll still get a little bit, even with just PFA and no detergent, you'll get some internal, but it's more like, do you see a significant difference between with the detergent or without? And if you do, then then you could make a more of an assessment about how much is truly cell surface versus just internalized.
Yeah.
You'll definitely still see some, even with just PFA fixation, that are internal. It'll just be a lot less.
Yeah. Yeah. But it'll be really nice to just validation and to try to distinguish those two. That's great. Thank you.
Hey, how's it going? My name is Iker. I'm at the Oklahoma Medical Research Foundation. So going off of Jason's point, one thing that you can do is you can label the outer segments with Cypher, which is a pH-sensitive dye. And therefore, it won't light up until it's inside of the phagosome. So even though the phagosome is not super acidic, it's acidic enough that it'll give you a signal. So that's one way to distinguish whether they're inside or outside.
Yeah. Does that work both? Have you tried that on? So is that that works in cell culture, I'm guessing. Do you know, does it also work? Would that also, well, no, I guess in vivo we're relying on the outer segments coming from in vivo. So we don't add exogenous. Okay. Yeah, it has to be an exogenous source so you can label it.
Has to be. Yeah. Also, like in work that we recently published, we also saw a similar thing where we starved the mice. And what we saw is that phagocytosis, at least measured in vivo, increased. However, interestingly enough, when I went back and tested this starvation condition in vitro conditions, whether it was RPE or microglia or whatever you wanted to call it, phagocytosis actually decreased. And for us, it led us to conclude that maybe it was increased in shedding of the photoreceptors, as opposed to maybe an decrease in phagocytosis. Do you have any idea? Exactly, yes. Your increase.
Yes. Do you know, we have, so our light onset is 6 a.m. So to be more, it would be, I need to wake up earlier and actually try this at the 6 a.m. time point, or even before light onset, maybe, to see if before that time where we think that the phagocytosis is happening. Yeah, I don't know other ways to really assess the increased shedding.
Would the photoreceptors be longer?
Because they're not being chewed up as much, or because they're synthesizing more?
Yeah, I mean, if they're not shedding their discs, then I would
Yeah.
assume that that material is still there. But that also assumes that the synthesis of discs is not affected by that.
It's not affected.
Yeah, my thought it would be that there'd be increased shedding. And that's why
Yeah.
she's the increase at 11 o'clock.
Yeah.
I guess I interpreted this as a delay, though. Not, I mean, we don't know one way or the other yet.
Yeah,
but-
exactly. There's so many, what's hard with interpreting this is there are a lot, you can't make strong conclusions on this because there are a lot of different things that could lead to this observation. If it were increased shedding, we don't, so with doing IHC and trying to do careful thickness quantifications, we haven't observed thicker outer segments, but I think that your ability to detect that is, you have to have a pretty dramatic difference to actually be able to say that. There is a difference. So there could be a subtle difference, or, 25% change that we wouldn't ever be able to probably detect just by, but we do actually, we do now have the capacity to do the OCTs in the mice, and we haven't done that yet in our diet animals. And maybe that would give us a bit more holistic view of that thickness. We've also talked about trying to do some tracing to look at lipid synthesis. Just because if you could rule out that lipid synthesis was increased, then you would probably assume that you're not having a huge increase in production of them. But yeah, what's shocking is there doesn't seem to be, we've done EM and there doesn't seem to be a buildup of debris like is seen in the MacTel patients. So whatever phenotype we're observing here, they are able to compensate for it, at least like well enough that we're not seeing dramatic phenotypes, like degradation or degeneration and loss of cells.
Sri has his hand raised.
Oh, I can't. Yeah, thank you.
Hi. So there is a possible way of dissecting out uptake versus binding.
Mhm.
maybe you can try surface biotinylation. So you feed the cells with outer segment, and after 2 hours or 1 hour to assess binding, you just move the cells to 4 degrees Celsius at which you would biotinylate, but you just biotinylate the surface, and then you can just look at total biotinylated protein in your 2 conditions.
Okay, that sounds, I don't totally, so you would so you biotinylate and it would only
You
get
don't fix it. You just
okay,
move the
so
cells
whatever
to
is on
4
the
degrees.
surface gets biotinylated because it won't get into the cells, and then you can quantify
How much is bound?
much. Okay, and should the biotinylation just get the outer segments?
I'll send you a
Okay,
protocol that you may adapt. I have done it with Xenopus oocytes to surface biotinylate.
okay,
protein.
fantastic. Thank you. I would really appreciate that. And
I'm also wondering what the rationale behind using dinosaur in this experiment is. Dinosaur is an inhibitor of endocytosis, but you are looking at a phagocytic process here, right?
Yeah, but I think they still rely on dynamin. So the phagocytosis still relies on dynamin, so it can work. We had started with dinosaur because we had used it as a positive control for our flow cytometry assay, because it blocked the ability for the labeled pus or the labeled outer segments to get into the cells. So we were kind of always using that as this positive control. We know like this will prevent them, and we can see what kind of our dynamic range is. And so we'd also tried it on this, not really with the intention of it. We actually thought we would see no bound, but it gave us something different, which then we interpreted as like they're stuck on the surface because they're not getting internalized, but they're getting bound.
The alternative explanation would be that by inhibiting endocytosis, you have just completely inhibited lysosome biogenesis. So the outer segment simply cannot get degraded and gets accumulated.
And they're stuck there. That's totally, yes. And we're doing it for 5 hours. So it's a pretty short. So I don't know how much, we can try a different time course too. Because within that 5 hours, there's like a continual, there's outer segments in abundance. So we think we would be able to catch. Yeah, but I guess, yeah, they're actively degrading them too. Yeah, that's a good point. Yeah, if you have, when you send the protocol, if you have a suggestion of other inhibitors to look at, that would also be fabulous.
Right.
I'd very much appreciate it.
I have a
Sorry,
random.
just to like help out here too. So we've used dinosaur before to inhibit phagocytosis in macrophages, and it does work. But another thing that you can also do is cytokinin D, which prevents actin rearrangements, and that also prevents internalization.
And it's a similar, will it catch them at a similar state? Like
Yeah, so
stuck but not internalized?
exactly. So they'll be bound but not internalized. And our treatments, we only do like 15 minutes before. So it's really acute.
Okay, and maybe that's what maybe we should try with dinosaur too,
Yes.
more acute treatment.
Actin, cholesterol, you can use cyclodextrin or cytocalicin D. They all should have the same effect. That
Okay.
actually truly inhibits uptake, unlike dinosaur, which also inhibits endocytosis.
The
Yeah.
turnover, okay.
I have
Fantastic, thank you.
a thought that's only vaguely related to all this, which I don't know, I guess we're all kind of speaking our minds. So if you have a disruption to sphingosine synthesis, and I think that's probably something you said you're going to look at,
Mhm.
then would it also be disrupting the formation of lipid rafts and thus the components that would be, I mean, maybe this is like your thought process already. It kind of seems maybe, but you know, if like you don't have the arrangement of proteins that you need to properly take up the outer segments, then that might explain a disruption or a delay, if it is in fact a delay.
We do, that's exactly what we are like, so one of the, I won't go all the way back, but one of the main things that we're seeing that serine is vital for is making sphingolipids, and sphingomyelin is one of the, is a main, like the bulk of sphingolipids is sphingomyelin, and we do see that reduced in both of our, like in our patients and also of our mice and our cell culture. And it's a major component of the cell membrane. And so we do think that we have, like, that is our main hypothesis, is probably there's something going on with the actual lipid function of the cell membrane. And then, yeah, RJ, who is here, has been trying a lot of different, we were trying like MERTK, things that depend on lipid rafts, and seeing if we saw less of it. And we're not seeing less of it. And the assays to actually visualize lipid rafts are pretty tricky. Or these like these lipid rich or cholesterol rich regions of the cells. But that is that, and then also the glycosylated sphingolipids, which are all really important for cell signaling and receptor. We do think that is something, but we haven't been able to like find one and pinpoint it and see something that we could follow up on.
What if it's not the abundance of a component in the membrane, but the organization of those components, or like the relative positions of multiple things that might normally sort of co-localize?
Yeah.
And I don't know
yeah.
exactly what you would look at, because that's not where I
Well,
don't know anything about it.
Yeah.
this is Jason. I mean, I actually, I realize we're derailing. So Mari, you should just tell us to shut up.
No, this is exactly what I was hoping for. So please derail.
I think this is part of the reason it would be so important to try your hardest to figure out if this is a binding event versus early internalization event, because I mean, you know, you guys have painted a nice story too about deoxyceramides and ceramides, which we know affect membrane curvature, especially at the point of endocytosis, but may not affect the initial binding, you know. So, you know, if this is truly an early biophysical curvature event problem where, you know, the signaling happens, but you just can't pull it into the cell initially, versus you can't actually organize the signaling at all because of raft content differences,
And your
etc.
receptors.
That, I mean, that really gives you, you know, the experiments to tell you if it's truly binding on an early internal event, I think points you towards, you know, one or the other possibilities that you can then explore in more depth. So that's why I do, even though it seems somewhat academic initially, I do think figuring out if it's truly the initial binding versus, you know, the beginnings, the uptake, is probably actually pretty important for
Yeah.
a mechanism.
Yeah. And it just gives you more insight, whether or not both of them are impacted by the sphingolipids, which they likely are. At least we can start narrowing down what we should be looking at. Yeah, this is great feedback. Thank you. So yeah, just to summarize this first part, the RPE seemed in our bulk RNA-seq, those RPE seem to be very deranged or changed from the serine and glycine free diet in the mice. And the transcriptomics suggest phagosome and lysosomal pathways. And as we've talked a lot about, our different assays give us some insight, but we really want to get into some of the details of that. Is it reduced binding or uptake? And then there's also, because the transcriptional pathway did suggest there was internal issues going on. It's not just the genes, at least, were involved in lysosomal function. So it's also possible that there's multiple things being hit that we can start to tease apart. And then we also are trying to see if we can find any phenotype that is a consequence of this altered phagocytosis. It's shocking to me that the MacTel patients have this panretinal defect, yet seem to be functioning fine everywhere but their macula, which it's almost unbelievable that you would have this debris just sitting there and a totally functional retina. So the eye's ability to deal with poor phagocytosis seems to be good in that situation and potentially in this model. But we know from other models that if you interfere with that turnover, it leads to degeneration of the photoreceptors. So it'll be interesting to follow up with. Thank you guys for your feedback on that. With the, what time are we at? So if there's a little bit more time, will also want to touch on this pathway that is whoops, I'm going to close the windows. The ones that are related to the metabolic state of the cells. And so if you look closely, I've zoomed in on the pathways. There is carbon metabolism, which is largely glycolysis, then the TCA, and then branched chain amino acid degradation. But then also fatty acid metabolism, which is again largely looks like it's fatty acid oxidation. So in looking at the pathways and playing with the genes, we came up with, the TCA cycle, it's like every enzyme of the TCA cycle. And these actually, the direction of these they're increased at the transcriptional level, all of these pathways. So just to like show a scheme of how the different genes we're seeing and serine glycine metabolism fit together. So glucose can come in, be converted to acetyl-CoA, and then feed the TCA. Serine synthesis is a branch off one of the glycolytic intermediates. So it takes 3-PG and uses that to build serine and glycine. And then a lot of the genes in the fatty acid metabolism and branched chain amino acid metabolism are oxidation and looking at the breakdown of these as substrates to actually as an alternative fuel for TCA. So one hypothesis we had was that in our mice with the serine and glycine free media or free diet, these animals are in a low serine and glycine condition. And so therefore they're going to increase their synthesis of serine and glycine and then pull carbon out of glycolysis. And this might reduce flow through glycolysis to some extent. And maybe as a way to compensate, then the cells increase oxidation of fatty acids and branched chain amino acids and potentially try to increase their TCA to compensate for it. So again, we used both in vitro and in vivo, and I have them in different order this time, but those Lonza HRPEs differentiated and then moved into a serine glycine free media. And then we did a glucose trace on those to start getting some insight. And so looking at glucose and the synthesis of serine, so to get you oriented here, we have cells that are in control media and then cells that are in serine glycine free media for 3 days or for 2 weeks. And the first thing I'm showing is here is the abundance. So the amount of serine that's present in, this is intracellular. And you can see that the cell's amount of serine drops dramatically. So the media is largely accounting for the majority of the serine in the cells. The labeling goes up a huge amount as a consequence of this. But if you actually correct the abundance by the labeling, so the labeling goes up because your pool size is so small here. So the amount of it that's coming from the glucose is relatively dramatically increased. But the actual flow from glucose to serine doesn't seem to be changed in these cells, which isn't what we were expecting. We were expecting this to go up. But you can see that they aren't really able to compensate for the loss of serine in the media.
Is PHGDH activity just like maxed out or something along that path?
Yeah, I'm going to get to that. So that's where this ends, is like, why is this happening? And in these cells and in vivo, PHGDH expression is very high and the protein is very abundant. So at least it seems like the protein level shouldn't be restricting it for PHGDH. So yeah, actually, I'll quickly.
Oh, I see where you're going. Okay.
Yeah, just going down though from here, pyruvate is down, lactate is down. Their labeling is somewhat increased again, probably because there's less of it. And then in these cells, we do see, so this is the TCA cycle. Many of the TCA metabolites are reduced both in their abundance and they're also reduced in their labeling. So it does seem like there is a consequence of the serine glycine free media on the downstream things. So they are linked. How to interpret it, we don't know just yet. We also, so the cells, we grow the cells or differentiate them in a very FBS low media. So it's 0.8% for the differentiation. So we were wondering, this isn't a very physiological level, and like normally cells would have a lot more access to other nutrients. So maybe this was a cell specific. So we also wanted to kind of see what was going on in vivo. So we returned to our model of the serine glycine free diet. And then to your point, Dan, so we looked in vivo and we haven't looked in vitro yet if the protein of PHGDH goes up, and fitting that model where you would have increased flux there, the protein level, it doesn't go up a huge amount, but it's a subtle and reliable increase. So there at least is some ability to increase the protein level some. And then we did the same trace. So a glucose infusion, 2 hours in the animal, and we were looking at serine labeling. And now this data is looking at the different tissues and either the control or the serine glycine free. So you can see in plasma, the mice fed the serine glycine free diet, they are not increasing globally, like the liver and the kidney don't appear to be increasing their serine synthesis at this time point. We did them in a fasted state. The retina, the neural retina itself has a modest but significant increase, and the RPE choroid did not. So fitting what we saw in cell culture, there is not compensation for the low serine and glycine that's being provided exogenously. Like they're not compensating for that. So yeah, this is your question, Dan. Is PHGDH just maxed out?
I have another question then. What about amount of serine in these tissues?
So the amount of serine, so
With a liver, you know.
we did this in a fasted state. So we have found that, so glycine, independent of time of day, fed or fasted, glycine is always quite low. Serine is not that way. So serine in a fasted state does not, like in the minus serine glycine diet animals have the same levels of serine as the control diet animals. And so this was a recent Rabinowitz paper. They also looked very closely into this in the plasma and the liver, same model and glucose trace, and they did glycine trace. And they said that their finding was also that synthesis of serine from glucose was not compensating for the diet change. Their theory of why the serine levels are maintained in a fasted state is that the catabolism of serine is reduced. So they're just using up less serine to maintain their serine levels. We, while in the kidney and the liver and the retina, so they're able, in the fed state, that's not the case. In the fed state, it drops, but that is because it doesn't have to do with the synthesis of it. It has to do with the fact that they're not actually just getting in more as they're eating, is the thought. But we do see a compensation in the retina, just not in the RPE choroid. And so our current hypothesis for this is not necessarily that PHGDH is maxed out. Actually, we had thought about this before, but at ARVO, we were fortunate enough to attend Lydia Finley's talk at UW, and she studies metabolism in stem cells and in cancer biology. And one thing she had said really stuck with us, in that to test if a cell is in a good redox state, they put it in a serine glycine free media, and if it can grow, then it can synthesize its own serine, and it's has sufficient redox state to allow that. If it doesn't grow and they can't proliferate, then it means that they're redox stressed. So it made us think of our RPE and wonder if that's what's happening. So to make serine from glucose, you need an NADH and it gets reduced. So to actually have sufficient flow through here to make serine, you need to have a strong NAD plus pool. And we think maybe that's what's limiting our RPE cells, and it's not necessarily pH-GDH that's maxed out, but the redox state of the cell, that they're not, and especially if the TCA cycle is not flowing to full capacity. So this is our current hypothesis, is that it's a redox issue. That is why the cells aren't able to compensate and make more serine in a serine depleted situation. This is the last data I have is, so to kind of get an insight into that, we did look at the pyruvate and the lactate levels in the cells. So this is the abundance in pyruvate. This is not significant, but there's a trend for pyruvate to be reduced in the serine glycine. This is all back of the eye, that RPE choroid. And then the ratio of the lactate to pyruvate is less favorable in the serine glycine. So maybe the little bit that they're trying to do is throwing off the redox and they can't do anymore. So that's our hypothesis right now. So yeah, the main finding when we did the tracing in vitro and in vivo is against what we were thinking. The RPE don't appear to be able to compensate by increasing serine synthesis to having reduced serine levels, kind of making it more of an essential amino acid to those cells. They're very reliant on what's coming exogenously. And so, yeah, we're hoping to test if it's the redox state that is limiting this. And then also, it's not clear why the TCA is down as a consequence of this. So we're going to look to see if there's a trend for it in vivo. It wasn't significant, but there was also this trend, alpha ketoglutarate was significantly reduced, to look at the mitochondrial content of the diet and the controls in both in vivo and in vitro, and see if they have reduced mitochondria. And they're trying, for whatever reason. And that's also worsening this redox state. So yeah, with that, I want to thank, I just have put here the people who've helped substantially with this work. So Roberto Bonelli works with us, and he's a biostatistician, data scientist. And so he did all of our RNA-seq analysis and helped us interpret that. RJ and Caleb, I think they're on this call, have done all of the hands-on work and both the cell and the mouse data. And then we work very closely with the Metallo lab, and both Emmeline and Courtney helped us with the measurements, the tracing measurements that I showed here. Courtney did the in vivo and Courtney did the in vitro and Emmeline did the in vivo. Yes, and with that, I will take questions. Dan.
So I assume that your first test of this redox situation is going to be like adding pyruvate and just seeing how your glucose tracing looks.
Yeah, exactly. That is in vivo. Adding some pyruvate was our first thought.
Yeah, I mean, I think that makes sense to me. But I think that there shouldn't necessarily be any confusion about why the TCA labeling would be less. If you're linking this to redox state, then wouldn't you think that too much NADH would sort of push back against the generation of more NADH in the TCA cycle?
That's true. That's exactly right. So it's the OXPHOS. So it's the electron transport chain that would be running. So yes, that's a very good point. I was compounding those in my head. The TCA cycle, yeah, they should reduce it and they should increase OXPHOS to help, like,
Dispose of it.
So why OXPHOS isn't, maybe it's not able to increase, but you're right. They don't want to produce more. And we do see, and I didn't put this, an increase in alanine labeling, suggesting that pyruvate is even being removed to make alanine, which doesn't make a ton of sense why it wouldn't be going to lactate. But
Maybe
I'll have to
have
look
extra
into-
nitrogen around, you know,
yeah,
because you're not making serine and glycine.
but they're making it at the same rate as they did before.
But the magnitude of, yeah, you're right. You're right.
So they
Um,
should be, and it's really
I see
they're
alanine as like kind of a relief valve a little bit, like not,
yeah,
it's not like it's energetically costly to make.
no.
And I just have one other thing, which is kind of a recommendation, but I usually have much more consistency when measuring lactate if I look at the rate at which cells generate lactate via an enzymatic assay.
Uh-huh.
So like just collecting media samples over time, and I can send you details on that if you'd like.
Yeah, you have sent me some other, yeah.
Or
So
I don't have to send it again.
no, I think I would like it again. But yeah, looking at the rate of lactate excreted from the cells or secreted, and then looking at that over time.
Mm.
Yeah, we have access to a YSI, which just directly measures, it's one of those membranes that has, it can measure glucose, lactate, glutamate, glutamine. So yeah, we could do that in media over time. That's a great idea. Do you have any thoughts for in vivo?
I mean, in vivo, how do you do anything over time like really
Yeah,
well?
that's true.
At least relevant to the retina.
Or the other thing the lactate to pyruvate ratio, like other ways to assess the redox state is also tricky just because we haven't attempted to measure NADH and NADH or NADH plus directly because of being told that it's so hard to like get a true readout of the redox state with those.
Even if you got a true readout, I'd still think that you'd be mixing compartments, and a
Yeah.
lot, like a lot of the people like Christian Metallo, I don't know if he'd be like grumbling about that because
Yeah, he doesn't look at
he doesn't what?
he doesn't look at NADH, like they
Yeah,
don't use
he
that
probably doesn't believe it because he's one of the
Yeah.
people who designed like a tracer just to look at, like
redox.
mitochondrial NADH versus
Yeah.
cytosolic. I mean, that's like maybe you can talk to him about tracer strategies actually,
Yeah.
because that four that do glucose that's deuterated at like the 4 position or something, that's really cool for getting NADH into the or yeah, for getting label on NADH in the mitochondria.
Into the minor.
But actually no, that's not perfect. I'll think about it.
Okay, thank you.
I
Francis,
mean,
yeah.
a quick question on did you measure what your medium, or what did you put into the medium in the first place? So I understand that there was very little serum. Did you analyze what the serum contained in terms of serine, glycine, glucose, glutamine and stuff?
No, the 0.8% FBS that we used, we didn't analyze it. We kind of, which was, it's actually helpful that that protocol had little serum. We have created like a KSR, like a serum replacement that doesn't have serine and glycine, because most FBS does have some levels of serine and glycine. We didn't check the 0.8%. So there is definitely some serine and glycine coming from that little bit of serum. But I don't know the level.
Then how much glucose did you put into your medium for your cell culture experiments?
For the cell culture, I believe it was 25 millimolar,
Okay.
but it might have been 12. If RJ, who's here, he did those, he could, I think so. What is so we were in DMEM F12 light conditions. And I believe that media, is it 25?
DMEM F12 is usually sort of halfway because the F12 doesn't have it.
Oh, that's right. Okay.
So it's usually halfway. It's like higher
Like
than
18.
physiologic, but lower than the DMEM, pure DMEM.
Yeah. Does that, yeah,
Okay.
what were you?
No, the question would be, or we've recently found that the retina actually consumes, the full retina consumes a lot of glucose, much more than we had thought. You know, we have similar glucose levels in our serum-free medium, like 19 millimolar is the normal level. And within 2 days, most of that is actually gone.
Yeah.
And then the question would be, what other potential substrates for energy metabolism are in there, like branched chain amino acids, glutamine, glutamate. Do you put any of that in there?
Yes. So the media, so the tracing media, so the normal media actually has pyruvate. It has glutamine, like the normal, what is it, 2 millimolar. It has the branched chain amino, so it has the essential amino acids. It doesn't have alanine. And then for the tracing, we keep all of that in there, except for for this tracing, removed glucose and pyruvate. Because we didn't want to dilute our pool with pyruvate. And then in hindsight, we're like, oh, maybe the pyruvate helps. But we haven't done that yet. But yes, it's a complete media in the amino acid. And so it doesn't have much in the way of lipids because of that very low FBS, but it's got glutamine, glutamate, and then the branch chains.
Okay. Now, and this question goes in the same direction, or question, as more a suggestion, I would say. It goes in the same direction as what Dan said. If you had the opportunity to do a metabolomic analysis of the medium before incubation, you know, the normal medium, the fresh medium, and then the spent medium, to look at what was consumed, and that versus your control medium with serine and glycine, and the medium without serine and glycine, what was consumed? Is there any change in what was being consumed that might give you hints as to what metabolism is going on? And at the same time, you know, if it's a metabolomic analysis, you know, you would also get lactate and pyruvate and other alanine, other interesting metabolites in one go.
Yeah, I think we actually did collect. We usually always collect the medium and keep some of our input media. We haven't analyzed it, but I think we have it. We can go back, but I think we'll also want to try it with more pyruvate. So we will keep it there, or we will definitely keep and analyze the media then. So that's a fantastic idea to pay attention to that. In the past, different models we've done, we actually saw a much greater change in serine. If we looked intracellular, the labeled serine wasn't great, but if we looked extracellular, we actually saw serine being secreted from the RPEs. So it's possible that we're losing a lot of our signal to the media that we're not appreciating in this assay.
That's consistent with your in vivo data as well, right?
Yeah. Actually, that does seem like a huge oversight now that you're like, why haven't we looked at the media? Yeah, that is a great point. Thank you.
Mari, I was just gonna, I mean, the concept I think you're introducing is the same. I mean, I think people are calling it reductive stress, right? That there's not enough sort of NAD to run things forward. So this is a generic suggestion, but if you can do a small amount of mitochondrial uncoupling in a non-toxic way, which, you know, you can push forward, you know, NADH consumption. So we've been using, and not because we found it, but others had found that BAM15 works very well in a non-toxic way compared to FCCP.
That's a fantastic.
And without a reduction in tier or toxicity, if you titrate it, you can email us afterwards. And, you know, then you could see, can you drive serine production
synthesis.
and that would help with the hypothesis that it's reductive stress, because you will inevitably be changing NAD to NADH ratios by doing that.
Yeah.
And I was also, I guess Dan was suggesting more pyruvate, but I could also imagine it's sort of in the opposite. If you put more lactate, do you get even less, serine synthesis because you keep,
Driving it the other direction.
driving it the other direction.
Yeah.
Well, that's kind of an issue that you're going to have to deal with the whole cell culture situation. If your RPE cells are consuming a lot of glucose and making a lot of lactate, you're dealing
Yeah.
with kind of a ramp in lactate throughout the experiment. And that's going to affect your ratio. So if you got to kind of make sure that your concentration of pyruvate, or if you add both pyruvate and lactate, which I would recommend just adding a really high concentration of both so that any lactate that gets made doesn't change the ratio too much. Then
In the media or in the cells?
in the media.
In the media. Okay. So you would actually add lactate to the media?
I'd add, I guess in my ideal experiment, different ratios of lactate and pyruvate. I've recommended this to other people, even on the call.
Yeah.
And the cells will generate lactate to some extent from glucose. And I guess the idea is just that you don't want the lactate levels or that ratio to be influenced too much by what the cells are doing.
Yeah.
So that's what you get by adding the exogenous lactate.
Like, to paraphrase, at least as Dan's portrayed it to me, was if you can get high enough levels of both pyruvate and lactate, you create a redox buffer system. And then the amount, like during glycolysis, the amount of lactate being produced is small compared to the overall buffer. So if you have a ton of lactate and a little bit less of pyruvate, that sort of theoretically fixes the redox state of the cell, favoring NADH. If you fix it with a ton of pyruvate and a little bit less lactate, maybe you fix it the other way. And if you start to see differences in serine production by like fixing the redox state of the cell, that could lend credence to the hypothesis.
Yeah, I had not thought of it that way. I'd only thought of like more lactate is bad. Like a very simplistic, like don't add lactate because then you want, but I see what you're saying, that if the media condition is such that the cell's activity during your trace isn't having a big effect on it, then you can kind of have this more fixed, steady state during the trace.
Yeah, like a
Or-
fixed redox state, exactly.
Fixed redox state.
Or you can just choose a really early time point and like not worry about the cells putting out so much lactate that it's making a gigantic impact.
Yeah.
Of one or the other. As
It
far
is
as
interesting. We've looked at a time course for serine synthesis with glucose, and it's been a while, so it's worth revisiting. But it did seem to take, so you get labeling of pyruvate and lactate within the first time point, the 15 minutes, but it takes longer for the serine labeling, which is also kind of curious when you think about that it's actually upstream of pyruvate and lactate in glycolysis. You think it would be a smaller pipe coming off, but it would still be a rapid pipe. So it's just and maybe it's a small enough pipe coming off of glycolysis that it takes time to see the accumulation of the label.
if the activity of the enzyme is really slow, but there's a pre-existing pool there, then glycolysis, the activity of all those enzymes is fast. You just can't even force that much water into the next pool,
Yeah.
if you're thinking about pipes.
Yeah.
Okay, I'm done. Other people should talk.
Yes, I really appreciate, this is exactly what I was hoping to happen. So thank you guys very much for all of the comments and suggestions. This is very helpful. And I'm sorry I took up your past the 9 o'clock.
Well, Nancy has a question, right?
Oh, all right. So Mari, really nice talk. Did you grow these cells on filters?
No, these were just on the plastic 6-well plates.
So maybe I would grow them on filters and really, lactate is high on the apical surface in the apical compartment of the RPE in vivo. And I think that really lactate is probably doing a lot for the RPE. So, I mean, I would even, and 12 millimolar glucose is just a lot compared to
Which,
in vivo.
yeah, I mean, what would be nice is if we could get our tracing window shorter so that we wouldn't have to flood with the glucose to make up for the end of the trace. I think that's because it's a fixed volume or a fixed amount that like they're burning through it quickly, you worry about them becoming hypoglucose towards the end of it, so you get that excess. But if we can reduce our tracing window, we could probably reduce our glucose concentration too. That's a great, and then yeah, putting them on the trans wells and getting them a lot more physiologically set. I'm sure RJ, like everybody's like, oh, not the trans wells. But it is a much better system. Yeah, thank you.
Okay.
Yeah, thank you all for this opportunity. Appreciate it.
Thanks for your
Thanks, Mari.
talk. We're gonna go on summer break after this talk. So look forward in the fall to a bunch more.
Fantastic.
And I'll stop the recording.